Welcome to this explainer. Right now, across the industry, there's this incredibly high-stakes debate going on. It's all centered around an irreversible biochemical countdown that gets triggered the exact second exsanguination occurs. We are talking about a rapidly closing, incredibly fragile window where muscle actually turns into meat, and literally every single second that ticks by dictates whether that meat is going to be a premium quality product or a complete commercial failure.
So the absolute first critical threshold we have to talk about is the 4 to 6 minute homeostasis collapse. Within just 4 to 6 minutes post-bleeding, the nervous system completely goes offline. The body's entire physiological apparatus for managing its oxygen, its temperature, its energy is just permanently lost. Without that blood supply, heat dissipation completely stops, and the carcass immediately starts retaining heat. And this isn't some gradual, gentle fade-out, It is an abrupt, total collapse of the system. You are suddenly left managing a closed biological machine that's locked into an irreversible decline. And that triggers an immediate chemical crisis. Because the stored oxygen is completely depleted, the body starts panicking, frantically trying to find energy anywhere it can. So it falls back on this rapid exhaustion of the creatine phosphate pool, desperately trying to rephosphorylate ADP back into ATP. But, here's the massive problem: that creatine phosphate reserve burns out so incredibly fast. The race against ATP depletion has officially started, and anaerobic resynthesis, it simply cannot win that race.
Which brings us right into the post-mortem pH cascade.
So what exactly happens when that initial energy reserve just totally runs out? The fatal failure chain kicks in. Since the aerobic pathway is blocked, energy metabolism violently pivots entirely over to anaerobic glycolysis. We're talking glycogen breaking down into lactic acid and 2-ATP. Now, in a living animal, blood circulation would just calmly clear that lactic acid right out of there. But in this closed post-mortem system, it's trapped. It can't escape. It relentlessly accumulates in the tissue, and that drives a catastrophic drop in the muscle's pH level.
To put that into perspective, look at the devastating initial pH drop for PSC, pale, soft, and exudative meat. It absolutely plummets. We're talking about a drop of roughly 1.6 pH units per hour in that critical first hour alone. That's over 5 times faster than the normal meat's drop of about 0.25 units. When pH nosedives that fast while the carcass temperature is still sitting super high, the muscle proteins literally denature right there.
If you trace that PSC trajectory, it's practically a sheer physical cliff right out of the gate. But then contrast that with the complete opposite end of the spectrum, DFD, or dark, firm, and dry meat. That line just stubbornly flatlines. DFD meat barely drops in pH at all because the animal's glycogen was completely zeroed out prior to slaughter, usually from stress or exhaustion. So we're really looking at 2 totally opposite biochemical failure modes here.
And make no mistake, these biochemical failures map directly to your P&L. PSC is, at its core, a rapid thermal processing failure happening right on the kill floor. It causes massive yield loss and purge because that denatured meat simply cannot hold its water. On the flip side, DFD is a pre-slaughter management failure that results in dark color and severely reduced shelf life because it totally lacks the acid needed to inhibit bacterial growth. Both of these are incredibly costly, and both are, frankly, totally avoidable.
All right, let's shift gears and dive into rigor mechanics, specifically when things get too hot.
To really understand the catastrophic impact of these thermal triggers on rigor, we have to talk about the ATP-MG complex. You need a very specific concentration of ATP complexed with magnesium to break those actin-myosin bonds and keep the muscle in a relaxed, extensible state. But as that ATP pool finally dries up, those bonds, they can't be broken anymore. Permanent, completely irreversible actin-myosin cross-bridges start to form, locking the contracting sarcomeres into a totally stiffened, rigid state.
And this right here is exactly why you've got 2 senior meat scientists debating the thermal impact of ambient temperatures above 20 degrees Celsius on rigor onset. It is absolutely crucial, because when ambient temperatures climb above that 20-degree threshold, your entire intervention window aggressively compresses. The heat drastically speeds up ATP depletion, which in turn violently accelerates that actin-myosin cross-bridging. So you aren't just dealing with normal rigor, you are dealing with a rapid, uncontrolled onset that locks in permanent toughness way before you even have a chance to properly chill the carcass.
Now, this rapid collapse happens through 3 distinct phases. First, you've got the delay phase, where the muscle is still relatively extensible. Next comes the fast onset phase, where that extensibility rapidly drops off. And finally, the completion phase, where everything is structurally fixed. But the absolute key takeaway here is this. The length of that crucial first delay phase is dictated entirely by how fast heat retention depletes your creatine phosphate. If your carcass is too warm, that delay phase basically just vanishes.
So, what does all this mean for the product itself? Let's talk yield loss and microbes.
Pivoting away from pure biochemistry to some pretty grim commercial realities for a second. Think about the number 2%. Just a 2% drop in water holding capacity is devastating. Why? Well, because as those actomyosin cross-bridges form, they physically squeeze the myofibrillar lattice, literally wringing water right out of the protein network. A 2% reduction might sound kind of small, but scale that up across hundreds of tons commercially, you're talking about massive tonnage lost to purge. You are literally watching your product yield drain away onto the floor.
And there's also a stark, urgent warning here regarding microbes, as rigor fundamentally alters these membrane properties and forces all that moisture to the surface. The body's natural defenses just completely shut down. It tears open this highly dangerous, unavoidable biological window for microbial colonization. You've got altered membranes, lots of surface moisture, and, especially in those high pH DFD environments, heavily suppressed bacterial inhibition. So, handling precautions right at this exact moment? They aren't just optional hygiene steps. They are absolutely critical interventions against a wide-open biological window.
Okay, so once rigor sets in, how do we get out of it? That's where enzymatic resolution and aging come into play.
So rigor is complete, but the process definitely isn't over. We now enter the highly controlled enzyme-mediated resolution of the rigor state. Remember that dropping pH we spent so much time talking about earlier? Well, as the tissue becomes acidic, it chemically triggers autolytic lysosomal enzymes, specifically cefapsins. These enzymes literally break out of the lysosomes and begin systematically tearing down both the myofibrillar proteins and the collagenous connective tissue.
If we look at the exact sequence of this internal dismantling, first, those altered membranes release the lysosomes. Then, the acidic pH activates those catheptic enzymes, and what they do is they target and rapidly disintegrate the Z-line structure, which is basically the main anchor holding the muscle fiber together. As that Z-line fragments, the whole myofibrillar lattice loses its rigid organization. The really important takeaway here is that aging is absolutely not decomposition. It's a highly precision, targeted enzymatic attack that results in true tenderization.
Which brings us to a rather provocative realization, one that completely changes how you view this entire process. Are quality failures, things like PSE, DFD, or toughness, are they really a chemistry problem or simply a rate management problem? Because here's the truth, the underlying chemistry is identical, and it's totally inevitable in every single carcass. The real, true measure of quality control is simply mastering the thermal rate of that cascade. If you control the temperature, you control the clock. So what steps will you take to manage the rate in your own processes? Thanks for joining me on this explainer, and keep questioning the science behind the systems.